Hey everyone. So we've been talking a little bit about DNA, but how do you replicate DNA, right? So we know a few things like you are an organism and organisms have offspring and organisms heal and repair and grow. And so that means we need more cells for that. And that also means we need more copies of DNA in order to fill those cells and give us all the directions we need to make and maintain these organisms. So let's look at the idea of how do you make a copy of DNA. So DNA replication has to be really, really high fidelity, and it is for the most part. And it follows what we now know as the semi-conservative model. It's important because for a long time we weren't really sure. Did it conserve part of the old strand, when you were making a new DNA molecule? Would you have, maybe a parent strand and then you just somehow made like a mirror image perfect copy, like non-conservative or, stuff like that. So we know now it's in this category here. So basically what happens are the two double helix strands separate. One of them becomes a template, right, or the example for the assembly or the copying of another strand. And that strand is going to be complementary, right? It's going to match up from all of these free nucleotides that are going to be floating around in the nucleus. We can use that. And then each new DNA helix has, again, the old strand right here, the parental strand, as well as that new strand. Okay? Right. So let's take a closer look. Something a little more step by step in this case. All right, so we have the parental, there it is. Parental molecule of DNA. And remember, it's double-stranded. Right? So double-stranded. So the first thing that's going to happen is it's going to unzip down the middle. And we actually use this really cool enzyme called helicase. Right? So that unzips DNA. After it's split into two, what happens next is we're able to use DNA polymerase, right? So these are, happy face. Well, that was, I guess, slightly grumpy. Okay. These two enzymes are called DNA polymerase. Okay. Notice the A's. That's how you know they're enzymes. So it's going to make a polymer of DNA, right? So here it goes, and then they're going to add on these corresponding nucleotides in each one of the new spots. So what ends up happening is we have two identical DNA strands, okay? So half of it's new, right? Half of it's new, and then half of it is actually from the parent. And so that's how we get that semi-conservative model. All right, because half of it, part of it, is conserved, right? And that would be how we kept part of the parental strand in there. Here's an even nicer looking graphic. Okay, so this is true to form for that helical structure of DNA. So here's that parental strand again, right? Here's the parental strand. First thing that happens is we get DNA helicase, right? So DNA helicase is going to come through and unzip these genes. See how it is? And then the next thing that's going to happen is we're going to get another, right? So DNA polymerase that's going to come through and add on these new nucleotides, right? And so C's are going to get G's, A's are going to get T's, and so forth and so on. So we keep that equivalent distance across the two strands to the two backbones of each DNA molecule. We conserve a parent strand in each, and we add a new strand, right? So the parent strand is also known as the template, right? That's the template strand over here, okay? All right, I think that's pretty good. Okay, so does it all start in the same place, or do we just start at the beginning of one chromosome and go all the way to the end? Well, that would take a long time. It actually would take too much time. We'd never be able to repair a paper cut in that if we did things that way. So generally what happens is we have multiple what we call origin of replications. So these little start sites, which are really easy to kind of tear apart, okay? So they open, and then the helicase travels along the distance of the entire strand of the molecule, right? Of the entire chromosome. It does it until they actually meet, okay, and completely open up. The other thing that happens, and you probably notice this little gray line down here, right? Little gray line. Okay, so that is where we're going to start adding in the new nucleotides, okay? So we talked about the idea of the parental or the template strand. This is the daughter strand, so this is the new one that we're putting in. And so as you can see, this can happen really quickly. So if we just follow the helicase and completely, as we're going, replicate and put in that daughter strand, by the time they're even done traveling or opening up, they've actually gotten 2 new DNA molecules. So that's pretty nice. And then let's talk about, real quick, prokaryotes, right? Prokes. What do they do? So remember, they have a circular or linear chromosome. Okay, so, and they're different from eukaryotes because they only have one origin of replication where it starts to separate. As it separates, new nucleotides are added to either side of the parental strand until finally we have 2 complete circular molecules, right? We also call these plasmids. Okay. All right, so that's how it's done in prokaryotes and eukaryotes. Now let's talk about the directionality of DNA synthesis. So after we've split down the middle, helicased it up, how do we know which side to start on? Well, that's kind of easy. What's interesting is that DNA polymerase likes to grab on, oops, there we go, okay, likes to grab on to the 3' end and start adding on 5', okay? So it's actually moving towards the 5' end, okay? So it's going anti-parallel. One way to remember that is that you can always think of this like lower number as your starting point, right? So you have less to start off with on your template or parental strand, right? And then you're going to go ahead and move, follow your parental strand, follow your template strand to the 5' end, okay? So remember, we're going to add on a 5' match or complement as we go. Okay, so that's the directionality. So if here is the parent or template strand, and it's 3', 5', and then it splits, right? So this is, it's anti-parallel. So here's the 5' over here, and here's the 3' over here. We're going to add new nucleotides. It becomes a 5' going this way towards a 3' finish. Same thing over here. We're going to start at this 3', and we're going to add 5' until we finish up at another 3' end. See how that works? So DNA polymerase is going to add on 5' ones to the 3' end as we go. Okay, I hope that's helpful. Now let's talk a little bit more about this directionality of DNA synthesis. So again, just like a better picture I thought might be, nicer than the one I just drew. So we have a parent strand right here, and we have the daughter strand over here, okay? And so remember, we're working with a molecule, and that molecule is DNA polymerase, right? Here it is. And that's going to polymerize or add on nucleotides to that template strand after it's been opened using DNA helicase. So the thing is DNA polymerase really likes to work on 5' to 3'. Like that's just where its happy place is, okay? So DNA polymerase, DNA poly, only moves from a 5' to 3' building direction. So what does that mean for this like other side, right? Because if it's inverse, then how do we even get a polymerase on there? Well, basically we do it with fits and starts. So every time we get just enough space unzipped from the helicase, we take a polymerase, prime it, and sit back down, and then try to just like catch up moving this way. So it means we have 2 different types of strands. One that's continuously transcribed, so this is known as the leading strand right here, Okay, that's the one that DNA polymerase is like designed for. And the other one is called the lagging strand, meaning it's kind of working against what it wants to do. It takes a little bit longer and there's some more steps to it. Like you have all these little nicks in the middle, okay, right? So look at these openings right here and here. Those little nicks have to be closed by another enzyme, DNA ligase, comes along and ligates or joins these two loose ends together. Why does it do that? Some people think it's because DNA polymerase evolved in in prokaryotes, right? And so remember, if they have the circular DNA, it's pretty good to just go all the way around one direction and the next. But since we have now, at least us eukaryotes, evolved, not a great way, but still a way to synthesize DNA using that lagging strand. So it's kind of a neat little evolutionary hangover that tells us about our story and our path of how we evolved to be who we are today. Some neat stuff. Okay, well, I hope that's helpful. If you have any questions, feel free to email me, and otherwise, I hope you have a great day.